Hi everyone, we are starting another chapter, so we are starting chapter 25, which is going to be on the urinary system. And in this video, we are going to cover anatomy. So we're going to look at the gross anatomy of the kidneys, microanatomy of the kidneys, and then gross anatomy of other urinary system structures such as ureters, bladder, and the urethra. It's going to be really important that you feel comfortable with the anatomy of the urinary system, especially the microanatomy, because this is going to become very important as you move on into further topics, such as renal physiology, which will be the next set of videos that you have. The organs that are going to be found in the urinary system include the kidneys, which are going to be the organs that produce urine, the ureters that are going to be the tubes that drain that urine away from the kidney down to the next component, the urinary bladder. The urinary bladder is going to be a temporary storage place for urine, and urine will be transported out of the body through the urethra. We're going to look at these organs in more detail as we go through this video, and we're going to start with your kidneys. The first organ we're going to look at in detail are your kidneys. Every day, your kidneys filter about 200 liters of fluid to remove toxins, wastes, and drugs from your bloodstream. They also, though, have other functions. They are going to work to ensure that the solutes that you need in your blood are returned to your blood and not lost as a waste product in urine. And they also are going to play roles in maintaining homeostasis. They regulate the total volume of water in the body and then the concentrations of solutes in that water. They regulate the concentrations of various ions in your extracellular fluid, and they work to help to maintain long-term acid-base balances. You may see a lot of these functions in great detail in some of the further physiology videos that will be posted. But today what we're going to be looking at is the anatomy of these structures. So get the anatomy down before you move on to start learning about how those structures function. So your kidneys are considered to be retroperitoneal, and what this means is they are not going to be located in the abdominal cavity suspended by mesenteries like we saw for your small intestines and your liver. Instead, they are going to be located posterior to the peritoneum or the serous membrane that lines the abdominal cavity. So you find your kidneys posterior to that serous membrane, the peritoneum, and then they're also going to be close to your vertebrae. So they're retroperitoneal. They are going to be located about the level of T12 to about L5. If you, though, look at this figure, so you're looking at someone in posterior view, and if you look at your right kidney, the right kidney is actually going to be a little bit lower than your left kidney. So here's your right kidney, and if you look and kind of draw a line over from your left kidney, you can see that the right kidney is a little bit lower in the body than your left kidney. And this is because, remember, on the right side of your body, you have a massive liver that is going to take up a very large amount of room in your right side of the body, and so it's kind of crowding out your right kidney. The other thing that you have closely associated with your kidneys will be adrenal glands or suprarenal glands. These are going to be glands that are found just above your kidneys, so just kind of superior to your kidneys. They're kind of diamond shaped and they look like a blob of chewed up bubble gum. Also, if you look at your kidneys, they do have a characteristic shape. They do look exactly like a kidney bean, that you are going to have a convex lateral surface and then a concave medial surface. So I'm drawing you a little kidney here, and so you have a concave, so going in, a concave medial surface, and you can see that here. I don't know why my kidney just disappeared, but I'll draw another kidney. So you have a concave medial surface, and then the lateral surface would be convex, and so you would have a convex lateral surface here. On the concave medial surface, you are going to have a lot of structures entering or exiting the kidney along this concave medial surface. The ureter that will drain the kidney will be found there, and you will have renal arteries and veins that will exit or enter at the medial surface, and then lymphatics. And if you remember back when we talked about the lungs, you had a lot of structures that would enter or exit the lungs, and the space that you would have those structures enter or exit the lungs was referred to as the hilum. So it's the same thing for the kidney. That area on the concave medial surface of your kidney, where you have all of your stuff exiting or entering, is going to be the hilum of your kidney. This figure really nicely shows you how your kidneys are retroperitoneal. So you are looking through a section through the body. These are the kidneys that have been cut, and then we have a vertebra, so this is L2, and then you have this big space here. This is your peritoneal cavity. The peritoneal cavity would house a lot of your GI organs. They have been removed for simplicity in this view. But you have this kind of white layer here that I'm highlighting with the yellow. This is going to be representing the peritoneum, or that serous membrane that would be lining the peritoneal cavity. And then you can see how the kidneys are going to be located posterior to the peritoneum and then very close to your lumbar vertebrae. So let's look at some of the internal anatomy of the kidney. So here is a human kidney. It has been cut along its frontal plane. So you're looking at a frontal section through a kidney. And we do have some very distinct structures. The first structure that you should know about is your renal cortex. The renal cortex is going to be the most superficial region, and it's going to be this kind of light tan colored portion that almost looks like a rind to the kidney. So all of this area here is going to be your renal cortex. The renal cortex kind of has a greeny or granular appearance to it, and it is the most superficial area. If you look deep to your renal cortex, these areas that are going to be kind of a darker tan or a darker red color, this is going to be your renal medulla. So deep to your renal cortex is your renal medulla, and the renal medulla is going to be composed of these triangular or cone-shaped structures that are referred to as renal pyramids. So I'm going to highlight one renal pyramid for you. So here's a pretty good renal pyramid. The renal pyramids are going to be part of your medulla, and you have a bunch of different renal pyramids that are going to be separated from each other by extensions off of your renal cortex. And so these light tan areas between adjacent renal pyramids, these are just extensions of your renal cortex that separates your renal pyramids. So these are referred to as renal columns. So those light colors are going to be your renal columns. And if we look closer at your renal pyramids, these are going to be these kind of darker colored, triangular shaped or cone shaped structures. A renal pyramid is going to have a base that is going to be much broader than this apex, which is referred to as a papilla. So all of your renal pyramids will have a wide base and then a narrow papilla. And the papilla, or the tip of the renal pyramid, is going to be really important because you are going to have urine released from the papillary area, or the papilla, and then drained into a tube that is near your renal papilla that's called a minor calyx. So you have a minor calyx, or a tube approximately here, that the renal papilla of that pyramid will drain into. So we said on the other view of the kidney that you have your outer layer of your renal cortex, and then deep to it you would have your medulla, which would contain your renal pyramids. If you take a renal pyramid and all of the cortical tissue around it, so that means the adjacent columns, the cortex near the base of the pyramid, and then the next renal column. What I've just circled, that area of the renal pyramid plus a renal cortex and two renal columns, all of that is referred to as a lobe. So we have one lobe kind of highlighted here, and you should have about 8 lobes per kidney. The other important structure, if you look close to the hilum on the medial side of the kidney, so on this side, you can see that you have kind of a broad triangular shaped structure here that then leads to a narrow tube-like structure. This narrow tube-like structure is the ureter. So you have your ureter here, which we'll talk about in a little bit. But then you have this broad, kind of wide, cone-shaped structure, and this is your renal pelvis. I think of the renal pelvis as being a funnel, and so it is continuous with your ureter, and so your nice funnel-shaped renal pelvis is going to work to ensure that you funnel all of the urine produced by the kidney into your long and narrow ureter. Okay, so I had said that every single one of your renal pyramids have a tip or a renal papilla, and that renal papilla is going to drain into a tube called a minor calyx. So we do have to look at kind of the tubes that are going to be draining your papillae and then carrying it towards the ureter. And so these tubes are going to start with those minor calyces. So here we have a single renal pyramid, and you would have this narrow portion being the papilla. And this is a nice view of a minor calyx that you have here and here. You're looking at the minor calyx from the inside. It's been cut, but you can see it's going to be a narrow tube that is going to be draining a specific renal pyramid. So that's going to be a minor calyx. Each and every single one of your papillae are going to drain into a minor calyx, and from the minor calyx you are going to have multiple minor calyces joining up to form something called a major calyx. So you have multiple papillae and minor calyces that then join up to form a larger tube called a major calyx, and that's what we're looking at right here. You will have major calyces will then join to then drain their urine into your renal pelvis that we have right here. It's going to be your funnel that then leads into your narrow ureter. So if you are looking at this kidney and trying to trace urine flow through the kidney, you would have urine running from your renal pyramid to the renal papilla, and then it would drain into your minor calyx. From your minor calyx, urine will then move into a major calyx, and for when you have two of your major calyces merge, you will have urine flowing into the renal pelvis, that widened funnel that then leads to your narrow ureter. And so then this figure is showing you all of the same structures, but now it's giving you an illustration right next to a section through a real kidney. And the illustration might make it a little bit easier to see what's going on with your calyces. So you again have very nice and clear a renal pyramid, and then the tip would be your renal papilla. The renal papilla is then draining into this narrow tube. That's going to be a minor calyx. You have a minor calyx, minor calyx, minor calyx from multiple of your adjacent renal pyramids. They are all merging to then form a major calyx. So you have a major calyx here. You'll have multiple major calyces. So you have a major calyx here, you have another major calyx here, and they are all going to then drain their components into your renal pelvis that you have here. And the renal pelvis is going to be that funnel that then leads to your ureter. If you know that your kidneys are going to filter the blood, it should come as no surprise that you are going to have an incredibly rich blood supply to your kidneys and then draining away from your kidneys. You have renal arteries, and these renal arteries are going to deliver about 25% of the cardiac output each minute. So these are going to be very, very vascularized organs. You will need to know arteries and veins within the kidney, but the good news is the arterial flow into and the venous flow out of the kidneys are going to follow a similar pattern. So you can kind of learn the arteries and know that the veins running next to those arteries will have the same name. Okay, so we're going to look at the arteries that are going to be supplying blood within the kidney. And what you want to keep in mind is this is how the blood is going to reach your nephrons, which are going to be the functional units of the kidney. So why you need to worry about these arteries is this is what's delivering the blood to the kidney in order to be filtered. And then for the veins, this is going to be draining the blood that has been filtered away. What you want to keep in mind is it looks like it's a lot to remember, but this is going to be an example of vena comitatus again, where the arteries and veins are running together and they have the same exact name. So if you can identify your arteries, the vein running right with it will be sharing the same name as the artery. So you only have to learn one set and just know that veins and arteries running together will have the same name. So we're going to start by looking at the arteries, and then you just know that the venous vessels running next to that artery is going to have the same shared name as the artery it's running with. So blood is going to be delivered to your kidneys through your renal arteries. And we talked about the renal arteries when we were going over arteries of the body. They are going to emerge off of that abdominal aorta. And for the renal artery, you have running right next to it a renal vein. You have a really nice color-coded picture for you here that the renal artery is shown to you in red and the renal vein is shown to you in blue. But there is a trick on being able to identify it, especially if you were looking at, let's say, a dissected human kidney. And that's in the walls of your artery versus the vein. The renal artery is always going to be nice and open because they have a little bit of bounce to their vessel walls. You can squeeze the renal artery walls together and then they bounce right back. And they're trying to show that to you in this picture, that you have this nice open renal artery right here, and it's next to your renal vein. Renal veins are going to be a little bit more fragile looking, and if you squeeze those walls together, they don't bounce back. So it kind of looks flimsy in this picture, not nice and open, and that's a good way of being able to tell if you're looking at renal artery or renal vein if you don't have the fake colors added to an illustration. But anyway, from your renal arteries are going to enter into the hilum of your kidney. So you're going to find them at that space with your renal vein and the ureter on the medial side of the kidney. Shortly after entering into the hilum of your kidney, your renal artery is going to branch to form 4 or 5 segmental arteries. So they're showing you 2 segmental arteries here. This would be one, here's another one, but then you would have one right here, another one over here. So you have 4 or 5 of these segmental arteries that branch off of your renal artery shortly after it enters into the kidney. So you go renal arteries to your segmental arteries, and branching off of your segmental arteries, you will have multiple interlobar arteries. So your interlobar arteries are going to be found in the renal columns, basically, or running towards those renal columns. So you would have an interlobar here, and then you can find it running up those renal columns. Here's another interlobar, and it's making its way to the renal column. Another one would be here, and they would be running with your interlobar veins. Then we get into our next artery. Your interlobar arteries are going to arch around the base of a renal pyramid. And when those interlobar arteries arch around the bases of your renal pyramids, they have a name change to arcuate artery. So think of it as it's this curved portion or this arched portion that's running around the base of your renal pyramid. So it is your arcuate artery. And basically, the arcuate arteries are going to be found at the junction between the cortex and the medulla. So you can use that arcuate artery as a dividing line that tells you anything deep to it is going to be medulla, anything that is superficial to it is going to be cortex. So that's a good way of kind of finding your dividing line between medulla and cortex. Then we get to I think a pretty artery. So you have all of these little squiggly lines that are kind of radiating off of your arcuate artery. And I can kind of draw it out for you. So you have this curved portion that would be your arcuate artery. And then in the cortex, you have all of these little arteries that radiate off of that arcuate artery. And so that's one of them right here. You have another one here. Those radiating arteries that kind of look like when you used to draw a sun and draw the rays radiating off of the sun, those radiating arteries coming off of your arcuate artery are your cortical radiate arteries. So they're cortical because they're in the cortex. They are radiate because they're like the radiating rays of the sun that you used to draw, and then they're arteries. What's neat about the cortical radiate arteries is if you look close, you have these little kind of arteries branching off of all sides of your cortical radiate arteries. So each and every one of these little branches is going to be a afferent arteriole. So you have afferent arterioles. So off of one cortical radiate artery, you have multiple afferent arterioles. And this is really important because it's going to be your afferent arterioles that are going to be delivering blood to the functional unit of your kidney, the nephrons. Okay, so that is kind of it for gross anatomy of your kidney, though you really can't see like afferent arterioles. But now we're going to move into some more detailed microscopic anatomy, and we are going to be looking at your nephrons. So I told you that the afferent arterioles that branch off of the cortical radiate artery, so here's an afferent arteriole here, they are going to be what delivers blood to your nephron. And so we're going to be talking about these structures that are shown to you in yellow. So there's your nephron. The nephron is going to be the structural and functional units of the kidney. They are going to be what actually forms urine. You have a ton of nephrons in each of your kidney. You have over 1 million per kidney, so millions of nephrons in your body. We are going to talk about the two main components of your nephron, and you can break them down into the renal corpuscle as well as the renal tubule. And what you want to keep in mind is as we move through talking about nephron anatomy, you should be asking yourself, what are the components of the renal corpuscle? What are the components of the renal tubule? What cells help to form those structures? And then also, what are the function of those structures? The other thing that you want to keep in mind is you really should know where they're located in the kidney. Are they located in the cortex? Are they located in your medulla? And a good way of determining that is to use your arcuate artery and your arcuate vein as your dividing line. So remember, your arcuate artery and vein are there we go, artery and vein, that's your dividing line between your cortex and your medulla. So anything in this direction is going to be located in the cortex. Anything in this direction is going to be located in your medulla. So you can use your arcuate artery and vein as a good dividing line to separate where's my cortex, where's my medulla, what's found in what area. And then just a review, because on the last slide it was not very clear when we were looking at the teeny tiny arteries coming off of the arcuate artery. So if this is your arcuate artery right here, you have this vessel branching off of it. That's going to be your cortical radiate artery. So you have the cortical radiate artery coming off of your arcuate artery. And then all of these little branches that emerge off of the cortical radiate artery, these are going to be afferent arterioles. So that's what I was trying to draw on the other figure. And then you can see that this afferent arteriole is then feeding into a nephron. You would have nephrons associated with all of these afferent arterioles. So you would have a ton of nephrons for just one cortical radiate artery. Okay, so let's just go back and look at your nephrons. So there are going to be two portions to your nephrons, the renal corpuscle and your renal tubules. We're going to look at the renal corpuscle first. And if you look at the renal corpuscle, you are going to have two components to it. So I'm going to circle, here's your renal corpuscle right here. And the renal corpuscle is going to have two structures. There's going to be a glomerulus, which is going to be this red structure, kind of that you see a ghost outline to because it's deep to that yellow structure. So in red, I'm circling your glomerulus. This is going to be a little capillary tuft. And if you look at the endothelium of the glomerulus, what you're going to see is it's fenestrated, or it has these really big pores or holes in the endothelium. This means it's super, super porous, and it allows for things to be filtered out of the blood as the blood moves through the glomerulus. So anything that's filtered out would be dumped into the next portion that we're going to talk about, the glomerular capsule, and that stuff that's filtered out is then referred to as filtrate. So the glomerulus is your first part of your renal corpuscle. The second part is all of this kind of round structure that's shown in yellow. This is your glomerular capsule. Think of this as being kind of a cup-shaped structure. It's hollow. It completely surrounds the glomerulus, and then it's continuous with the next portion of our nephron that we're going to talk about, the first part of your renal tubule. So as you have filtrate formed, it is going to then fill your glomerular capsule and be able to then enter into the next portion of your nephron, which would be your renal tubule. But let's look a little bit closer at your glomerular capsule. So this is still a portion of the renal corpuscle, but it was going to be that kind of cup-shaped hollow structure that surrounds your glomerulus. If you look closer at the glomerular capsule, you will see that there are going to be two distinct layers to it. There is a parietal layer. This is going to be the most superficial layer. So all of this is going to be the parietal layer. And the parietal layer of the glomerular capsule is going to be formed from simple squamous epithelial tissue. So you have simple squamous epithelium shown right here. The deeper layer is going to be the visceral layer. And the visceral layer is going to come into contact with the basement membrane and then your endothelium that forms your glomerulus. So this has to be deepest because it's going to come into contact with that glomerulus that's within the capsule. And if you look at the cells that make up the visceral layer, you're going to see some really weird looking cells. They're actually really pretty, but the cells that form your visceral layer are referred to as podocytes. And so these are going to be epithelial podocyte cells or podocytes. And what is special about them is you have these bizarre little foot-like extensions that emerge. So here's a little foot-like extension that I'm highlighting in red. Here's another one. And so you have these little foot-like processes that extend off of the cell and cling to the basement membrane. But because you have these little foot-like processes, it is going to create these little spaces where you don't have a portion of the cell. So this little space right here is going to be referred to as a filtration slit. And the filtration slit is going to be important because you have these podocytes closely associated with your glomerulus, and they have slits in the cell between those podocytes, and these filtration slits will allow filtrate that is passing through the endothelium of your glomerulus to pass through the filtration slit and then enter into the space of the glomerular capsule. But let's look a little bit closer at your glomerular capsule. So this is still a portion of the renal corpuscle, but it was going to be that kind of cup-shaped hollow structure that surrounds your glomerulus. If you look closer at the glomerular capsule, you will see that there are going to be two distinct layers to it. There is a parietal layer. This is going to be the most superficial layer. So all of this is going to be the parietal layer. And the parietal layer of the glomerular capsule is going to be formed from simple squamous epithelial tissue. So you have simple squamous epithelium shown right here. The deeper layer is going to be the visceral layer. And the visceral layer is going to come into contact with the basement membrane and then your endothelium that forms your glomerulus. So this has to be deepest because it's going to come into contact with that glomerulus that's within the capsule. And if you look at the cells that make up the visceral layer, you're going to see some really weird looking cells. They're actually really pretty, but the cells that form your visceral layer are referred to as podocytes. And so these are going to be epithelial podocyte cells or podocytes. And what is special about them is you have these bizarre little foot-like extensions that emerge. So here's a little foot-like extension that I'm highlighting in red. Here's another one. And so you have these little foot-like processes that extend off of the cell and cling to the basement membrane. But because you have these little foot-like processes, it is going to create these little spaces where you don't have a portion of the cell. So this little space right here is going to be referred to as a filtration slit. And the filtration slit is going to be important because you have these podocytes closely associated with your glomerulus, and they have slits in the cell between those podocytes, and these filtration slits will allow filtrate that is passing through the endothelium of your glomerulus to pass through the filtration slit and then enter into the space of the glomerular capsule. This figure really does a beautiful job at showing you the components of your renal corpuscle. So in red, you have your little capillary tuft of your glomerulus. So here is your glomerulus. And you can see that feeding into the glomerulus is the afferent arteriole. Draining the glomerulus is an efferent arteriole. And this picture doesn't really give it justice that the afferent arteriole is going to be a larger diameter vessel than your efferent arteriole, and that can be helpful if you are looking at an unlabeled figure to be able to determine which is the afferent arteriole bringing blood into the glomerulus and then which is the efferent arteriole that is draining blood away from it. So you have your glomerulus, that's one part of the renal corpuscle. But then we need to look at the glomerular capsule. And so remember, the glomerular capsule has two layers. You have a parietal layer, which would be this outer layer right here. So you have your parietal layer of the glomerular capsule, and then you have a visceral layer. And the visceral layer is going to be these weird looking cells, those podocyte cells that are going to be intimately attached to the glomerulus. So you have these funky looking podocyte cells with all of those little foot processes and then the little filtration slits. So you have your podocyte cells that are going to be forming that visceral layer. And you can kind of see it kind of hugging the glomerulus itself. So those are the two layers of your glomerular capsule. You do have this physical space between those two layers. And so this is the glomerular capsular space. And this space is important because as you have blood move through the glomerulus, remember that it has holes or pores that anything small enough can exit. And so what exits the glomerulus and goes through those little filtration slits is the filtrate, and the filtrate will fill in this glomerular space, this space in your glomerular capsule. And the glomerulus we're going to see is going to be continuous with the next part of your nephron, the first portion of your renal tubule, which is your proximal convoluted tubule here. So you can have filtrate in your glomerular space, and then it'll move out of the renal corpuscle and into the next component of your nephron, which would be a part of your renal tubule, the proximal convoluted tubule. On the figure to the right, they are just kind of zooming in to take a look at the relationship between those podocyte cells of the visceral layer of the glomerular capsule and the glomerulus. So you have your glomerulus, or the little capillary, and this is the endothelium. You can see that you have these holes in it. These are the fenestrations or the pores. And then you would have a podocyte cell that is going to be kind of wrapping around the glomerulus. And you have those little foot processes that extend off of the podocyte. And so you have these areas where you don't have the podocyte cell covering the endothelium. You can see the endothelium kind of peeking through, and where you have them peeking through, it's a filtration slit. So the filtration slit is going to allow for substances to filter through those pores or the fenestrations of the glomerulus and then fill that glomerular space. And if you ask me, the podocyte cells are kind of pretty. The second component of a nephron would be your renal tubule. So remember, you have the renal corpuscle and the renal tubule that will form a nephron. So here, these arrows are pointing to your portions that are the parts of your renal corpuscle. So all of this is the renal corpuscle, and you would have deep in red is the glomerulus, and then surrounding it in yellow was that glomerular capsule. So that would be the components of your renal corpuscle. You have your renal tubule formed by the remaining structures in yellow here. And so if we look at these structures, what we have will be three components to your renal tubule. The first part of the renal tubule is going to be continuous with your renal capsule, or your glomerular capsule, sorry, and that is this kind of very convoluted or twisty tube called the proximal convoluted tubule. So if you remember back to A&P1, proximal was used to describe the placement of a portion of the limb that would be closer to the root or the head than another portion. So in this case, proximal is being used in a similar fashion. It would be the portion of the renal tubule that's closest to your renal corpuscle. So it is going to be proximal because it's continuous with and closest to your renal corpuscle, but it is also going to be convoluted because it's kind of twisty. So you have a proximal convoluted tubule. The second part of your renal tubule is going to be this U-shaped structure here, referred to as the nephron loop. And then the last portion of your nephron is going to be another twisty tube, and this time it is further away from your renal corpuscle, so this is your distal convoluted tubule. The other important thing is you do need to kind of know where these components are located within the kidney. So you should be familiar with where the portions of your renal corpuscle will be, as well as your renal tubule. So this is where the arcuate artery is going to come into handy again. So you have your arcuate artery and vein, your dividing line between cortex, everything this way, and medulla here. And you know that the glomerulus and your glomerular capsule are going to be all located in the cortex. And if you look at the components of your renal tubule, 2 out of 3 of the portions of your renal tubule are going to be located in your cortex. So the proximal convoluted tubule is cortex. Your distal convoluted tubule is also going to be cortex. But if you look at your nephron loop, this is diving down into your renal medulla. So you would have your nephron loop being located within your renal medulla. All right, so we are now going to look at the 3 different regions of your renal tubule in more detail. I've already said that you should be aware of whether the portions are located within the cortex or the medulla, but then you should also be aware of what are the cell types that are found in each portion, and what does that cell type tell you about the function of that specific portion of the renal tubule. So we're going to start with the first portion of the renal tubule, the one closest to the glomerular capsule. So we're looking at your proximal convoluted tubule. We've already said proximal convoluted tubule is going to be located just in your cortex. And if you look at the cells of your PCT, or your proximal convoluted tubule, what you're going to see is the proximal convoluted tubule is composed of cuboidal cells that have a really dense collection of microvilli along their borders. And so this would be the internal border. So it would be lining the tube. This would be the external surface here. The microvilli are going to form a brush border. And if you remember back to when we were talking about the digestive system, we saw brush border for cells. And again, it was going to be a way that you were able to increase the surface area of, in that case, the small intestine, without making the volume that much larger. And so it's the same thing that you have going on in your proximal convoluted tubule. You have this dense collection of microvilli that forms a brush border, because this is a way to increase the surface area without making the proximal convoluted tubule have to be really long. If you think of the fact that there's a brush border with this dense microvilli, that makes sense that one of the really big functions of your proximal convoluted tubule is going to be reabsorption and secretion. So reabsorption and secretion is something that's going to go into nitty-gritty detail in the physiology lectures, but this is something that I find people have a hard time with. So whenever you think reabsorption, what you should be kind of thinking or saying when you see reabsorption is the reabsorption of water or good solutes from the filtrate back to the blood. So when you see reabsorption, you reabsorb from the filtrate back to the blood. And if you keep that in mind, it's going to help you when you're going through the physiology. So reabsorption means that you are going to reabsorb good stuff, water and solutes, from the filtrate back to the blood. You don't want to lose any good stuff, so you have to reabsorb it from the filtrate and send it back to the blood. So that helps me remember what reabsorption means, and I've seen a lot of people have trouble with what reabsorption means. So if you see reabsorption, you reabsorb from the filtrate back to the blood. For secretion, you're going to talk about this a lot when you are looking at the physiology and how these things work, but you want to think of, for secretion, it's the opposite. You are going to dump more substances into the filtrate. So it's adding substances to the filtrate, not removing it and dumping it back into the bloodstream. That's reabsorption. So your proximal convoluted tubule is then going to transition and be continuous with this U-shaped nephron loop. So the nephron loop, remember, is the only portion that is going to be located in your medulla. So you have your nephron loop within the medulla of the kidney, and you have two distinct arms to your nephron loop. So you have, if you follow, that filtrate would be running through your proximal convoluted tubule and then entering and then running down your nephron loop. That down portion of your nephron loop is referred to as the descending limb. So you have the descending limb, and it gets its name based on how filtrate is going to flow through the nephron loop. So filtrate is going to run down this limb, so that is your descending limb. And then filtrate is going to run back up this limb, so then this is your ascending limb. The descending limb, if you look at the cell type, it's simple squamous epithelium. And then if you look at your ascending limb, it kind of shows you on this figure, your descending limb was much thinner, and then you have a transition part way up your ascending limb that it is a thicker tube. And so this thick portion of your ascending limb, you're going to have cuboidal to columnar cells. It could be thin in some of your nephrons, depending on what nephron you're looking at. The nephron loop is going to be continuous with your last portion of your renal tubule, the distal convoluted tubule. So here you would have your ascending limb running up, and then once you get back into the cortex, you have this nice kind of twisty tube. That is your distal convoluted tubule. If you look at the cells of your distal convoluted tubule, they are cuboidal, again, just like we saw for your proximal convoluted tubule, but the big difference is there are very few microvilli on the cuboidal cells associated with your distal convoluted tubule. So there might be a few of your little microvilli, but you don't have that dense brush border that you saw on the cells for your proximal convoluted tubule. So this makes sense if you look at the function. The function of the distal convoluted tubule is going to be more involved with secretion, so secreting things into the filtrate so that it is removed from the body as urine, and it's not really involved in reabsorbing good stuff from the filtrate back to the blood. That's going to be really the job of your proximal convoluted tubule. So proximal convoluted tubule with the really dense microvilli, reabsorption is super important. Secretion is also occurring. And then if you look at your distal convoluted tubule with their sparse microvilli, it's going to be more involved in secretion than reabsorption. Just like your proximal convoluted tubule, your distal convoluted tubule is going to be located just in your cortex. We have one other structure to talk about, and these are going to be your collecting duct. So here is one large collecting duct, and you can see the collecting duct is going to run through the cortex as well as your renal medulla. And so the collecting duct is going to be receiving filtrate from multiple nephrons. So it's not a part of your renal tubule, but you would have renal tubules draining into it. So you can see here your distal convoluted tubule is draining into this collecting duct, but you would have a distal convoluted tubule draining into each of these cut portions right there. So you have multiple nephrons that will be dumping filtrate into these urine collecting ducts or collecting ducts. They do dive down into the medulla and they run down the length of your pyramids. And if you look at the illustrations of your kidneys, or if you look at a real one, they always kind of show that you have these little stripy patterns on your renal pyramids. You can actually see these on a kidney that's been dissected. These little stripies running down the length of your pyramids are actually the collecting ducts. So you have multiple collecting ducts running down your renal pyramids, and they're all running down towards the papilla, or the tip of your renal pyramid. And so this is how that papilla is going to drain that urine from the renal pyramid and dump it into your minor calyx. And if you look at the cells that make up the collecting ducts, there's going to be two different cell types that are going to be found. The first cell type is called a principal cell. Principal cells are going to have very few really short, nubby microvilli. And so they don't have many microvilli, and the ones that they have are going to be pretty short. But the principal cells are going to be important for maintaining water and sodium balance. The other cell that you have is an intercalated cell. And if you look at this, it's cuboidal in shape, but you are going to have much more abundant microvilli. And so you can tell it apart from the principal cell by the large amount of microvilli. There are two different types of microvilli actually found on these cells. And what the intercalated cells are going to help with is to maintain acid-base balance of your blood. So this figure is going to nicely summarize all of that information we just went through. So it's showing you the components of your nephron and the collecting duct. It is associating it with where they are located in reference to the cortex and the medulla. Don't forget about your arteries and veins that you can see in this view. And then they're breaking down the different kind of portions of your nephron. You have your renal corpuscle that's going to be including the glomerular capsule and the glomerulus. And then if you follow it out, it's kind of like picture in picture. Over here on the right, they're telling you the cell types for each component. So cells of each component. So if you remember back to the glomerular capsule, you had this nice parietal layer that was the simple squamous epithelium. In the visceral layer, you would have your podocyte cells. So this is a good way of being able to review. Do you understand what the different components are, where they're located, and what cell types are there? What's missing would be try to tie the cell types, tie it to function. And this is going to be something that you are going to talk a lot about the function of the different regions of the nephron in the future physiology lecture. But if you already kind of know that the proximal convoluted tubule with your little cuboidal cells with lots and lots of microvilli forming a brush border, that will help you to remember that it's involved in reabsorption from the filtrate to the bloodstream of good substances, and then it's also involved in secretion. It will help you to remember that for your distal convoluted tubule that doesn't have many microvilli, it is going to be primarily involved in secretion, and there's a little bit of reabsorption that occurs. So if you kind of summarize for yourself, learn the anatomy, learn the cell types, and then try to tie it to what is happening in the different portions of the nephron, that will help you to be able to grasp the physiology of the kidney. There are two different types of nephrons located in your kidney. The first type of nephron is called a cortical nephron, and that's what you are looking at here. And then the second type of nephron is a juxtamedullary nephron that you're looking at here. These nephrons are going to differ in their shear number, the length of their nephron loops, and also their functions. The majority of your nephrons are going to be cortical nephrons. So 85% of the nephrons in your kidney will be considered cortical nephrons. They get their name because they are going to be primarily located within your cortex. So remember, we can use your arcuate artery and vein as your dividing line. Then anything above the artery and vein in this direction is going to be in the cortex, and anything in this direction is going to be in your medulla. So if you look at your cortical nephron, the majority of the components of the nephron are located in the cortex. This is going to include your renal corpuscle, which would be your glomerulus and your glomerular capsule, and then two components of the renal tubule, the proximal convoluted tubule and the distal convoluted tubule. You do have your nephron loop extending down into the medulla, but that nephron loop is pretty short, and so it doesn't dive very deep into the medulla of the kidney. Cortical nephrons, their function is to deal with reabsorption of something good from the filtrate back to the bloodstream, and then secretion of something to kind of fine-tune what is going to be within urine. So secretion would be secreting something from the blood to the filtrate. You have cortical nephrons highly involved in reabsorption and secretion for their functions. If you look at your juxtamedullary nephrons, you can see that they have incredibly long nephron loops that dive really deep into the medulla. So this is going to be the big difference between your cortical nephrons and your juxtamedullary nephrons. This really long nephron loop that's going to deeply dive into the medulla. You also are going to have a difference in the function. Juxtamedullary nephrons, they are not as common in your kidney, so there's only about 15% of your nephrons would be considered juxtamedullary nephrons, but they have an important function. They are going to play a huge role in being able to produce a very concentrated urine, and this is important to prevent you from dehydrating. If you are dehydrated, you need to reclaim as much of that water as possible so that you aren't getting rid of valuable water as a waste product, and so your juxtamedullary nephrons are going to help play a role in being able to produce that concentrated urine. Your nephrons are going to be associated with a few different types of capillaries. We already spoke about one of them, the glomerulus. So remember that the glomerulus is a capillary tuft, and so that would be one type of capillary associated with your nephrons. So there's your glomerulus. Another capillary bed, though, that are associated with your nephrons are going to be peritubular capillaries. The peritubular capillaries are shown in purple on this figure, and it's all of this kind of webbing or network of vessels surrounding your components of the renal tubule. Peritubular capillaries are going to be involved in reabsorbing solutes and water from the filtrate back into the blood. And if we look at the peritubular capillaries, you can see that they are receiving blood from your efferent arteriole that is draining that glomerulus and then delivering blood into the peritubular capillary bed. And then the peritubular capillaries themselves will be draining into a venule that you see here next to the black arrow. And that venule will eventually drain into your cortical radiate vein that is found running next to your cortical radiate artery. If we look at your juxtamedullary nephrons, the ones with the really long nephron loops that dive into your medulla, you are going to see that there are going to be associated with specialized capillary beds called the vasa recta. So the vasa recta is going to be specialized. It is found in juxtamedullary nephrons, and it is going to be this kind of long, open webbing surrounding your nephron loops in your juxtamedullary nephrons. So your vasa recta is going to receive blood from the efferent arteriole. And so if you look here, you still have that efferent arteriole draining the glomerulus that's going to go and eventually give rise to the vasa recta. But when you look at the efferent arteriole draining the glomerulus associated with the juxtamedullary nephron, you don't have that arteriole breaking down into that kind of complex network that you saw with the peritubular capillaries in your cortical nephrons. So you have the vasa recta is going to be formed by these very kind of long straight vessels that run parallel to your nephron loop. The function of your vasa recta is going to be very different. So the vasa recta is going to be involved in forming concentrated urine. But just like we saw for your peritubular capillaries, the vasa recta will drain into a venule that will eventually drain back into your cortical radiate vein. We can look closer at these different capillaries associated with your nephrons. So the glomerulus, it is this specialized capillary tuft, and this is going to be specialized for filtration. We've already seen how they have the fenestrations or the pores that are not very small, and so if something is small enough to fit through that pore, it's going to be filtered out of the blood and then added to filtrate. So the glomerulus is going to be specialized for filtration. It's going to be the only place that you have filtration occurring in the kidney and in your nephron. The glomerulus, though, is a little bit different than other capillary beds that we've talked about, so it doesn't quite fit the same definition that we got when we were talking about vessels. And this is because the glomerulus is going to be fed and drained by an arteriole. So I've said in a couple of the other descriptions that you have an afferent arteriole with an A that is going to feed blood into your capillary. And so this is going to be this structure right here. Your afferent arteriole is going to feed blood into the glomerulus. It is going to be drained by another arteriole called the efferent arteriole. And that's going to be this one right here near that black arrow that I just circled. The efferent arteriole is going to actually be a little bit smaller in diameter than your afferent arteriole, which is a good way of being able to figure out which one you're looking at. You can look at the size. The larger diameter is going to be your afferent arteriole that feeds blood into the glomerulus. And then you would have a smaller diameter efferent arteriole that would be draining blood away from the glomerulus. Blood pressure in the glomerulus is high because of this. And if you remember when we were talking about vessels, arterioles are going to be high resistance vessels. And so you do have a very high pressure within the glomerulus. Peritubular capillaries are going to be low pressure and very porous capillaries, and this is going to allow them to be really involved in reabsorption of good solutes and water that you don't want to lose in your filtrate. So it's involved in reabsorbing water and solutes from the filtrate back to the blood. Earlier we talked about how they are going to arise from your efferent arterioles that are draining the glomerulus. So you can see that here. And they are going to be clinging to the renal tubules in the cortex of your kidney. And they are eventually going to drain into a venule that will then drain into your cortical radiate vein. And again, if you are looking at juxtamedullary nephrons and only juxtamedullary nephrons, you are going to have a vasa recta. The vasa recta is going to be these very long, thin-walled vessels that are going to run down the long nephron loops of just your juxtamedullary nephrons. They also are going to arise from the efferent arterioles that are draining the glomerulus. So you have your efferent arteriole here, and that is going to feed into your vasa recta, and it will be drained by a venule and dumped back into your cortical radiate vein. And the function of your vasa recta is going to be to form very concentrated urine. And you're going to talk about how that happens when you get to the physiology videos. All right, I promise this is the last component of a nephron we need to talk about. So every single nephron is going to have a specialized region that is going to be your juxtaglomerular complex. And this is going to be located where the distal end of the ascending limb of your nephron loop is going to come up against and lie against an afferent arteriole that feeds the glomerulus. Sometimes it might lay against an efferent arteriole, but most of the time it will be laying up against a afferent arteriole. And this seems weird, so if you look at the figure here, the picture might help better than having a verbalization of what this is. So here we're looking at a nephron. You have your glomerular capsule here, you have the glomerulus within it, and you would have an afferent arteriole, this would be right here, that would be carrying blood into your glomerulus. So there's your afferent arteriole. You also have your nephron loop that would be found right here. So there's your nephron loop, and you would have right here is your proximal convoluted tubule, and then you would have it being continuous with the descending limb of your nephron loop. So then running back up on this side, you would have your ascending limb of your nephron loop. And where the ascending limb of the nephron loop comes into contact, right here where the box is, with the afferent arteriole, that is going to be a specialized region called the juxtaglomerular complex. The ascending limb of your nephron loop and the afferent arteriole are going to be modified at this point of contact to form this juxtaglomerular complex, or JGC. All right, so here we're looking at a zoomed in view of the juxtaglomerular complex and then the renal corpuscle that it's near. So to orient, this region right here is going to be the juxtaglomerular complex. It is going to be that you have your afferent arteriole here. So your afferent arteriole, that would be then leading into the glomerulus that you see in the renal corpuscle in that glomerular capsule. But you have your afferent arteriole, and then this structure right here, this is the ascending limb of your nephron loop. So you have the ascending limb of your nephron loop here, and then you would have this point of contact between the afferent arteriole and then the ascending limb of your nephron loop. So then this really is that juxtaglomerular complex. The juxtaglomerular complex is going to contain special cells that are going to be important in regulating the rate of filtrate formation as well as the systemic blood pressure. And those three cell types are going to be the macula densa that will be found in the ascending limb of the nephron loop, granular cells that are going to be associated with your afferent arteriole, and then these extraglomerular mesangial cells that are going to be kind of between the two. So we're going to take a look at these cells, and then that is it for nephrons. The first type of specialized cell within your juxtaglomerular complex is the macula densa. So the macula densa is going to be this area right here. It is associated with your ascending limb of the nephron loop. And macula densa really means dense spot or dark spot. So it's going to be this specialized region where you have a series of tall, very closely packed together cells in the ascending limb of the nephron loop. This is actually going to be a point of contact between the ascending limb of your nephron loop and that afferent arteriole. Why the macula densa is important is this is going to be where the cells are chemoreceptors. And so they're chemoreceptors that are monitoring the sodium chloride content of your filtrate as that filtrate is entering into your distal convoluted tubule. The second special cell associated with your juxtaglomerular complex will be your granular cells. The granular cells are going to be found in the walls of your afferent arterioles. So you can find them here and here. So they're in the walls of the afferent arteriole. What these cells are actually big, enlarged, smooth muscle cells that will have these granules that contain the enzyme renin. These granular cells are going to work as mechanicoreceptors that will be able to sense blood pressure in that afferent arteriole. And last but not least, the last type of cell that we need to worry about for your juxtaglomerular complex are the extraglomerular mesangial cells. These are going to be located between your arteriole and the ascending limb of the nephron loop. So they are going to be these cells that are kind of filling the gap between those two structures. These cells are going to be interconnected via gap junctions, and they might play a role in passing regulatory signals between your macula densa and your granular cells. All of the components of your juxtaglomerular complex are going to be kind of dealt with in more detail when we talk about physiology. There's a lot of information in this figure, and there is a lot going on in this figure. But what you want to do is try to go through and see if you can identify first, start off with your components of a nephron. Where are the components that would be the portions of the renal corpuscle? Where would be the portions that would be portions of the renal tubule? Where are your cortical nephrons? Where are your juxtamedullary nephrons? After you tackle that, start to look at where are your collection ducts, what components out of your nephron and the collection ducts are found in the cortex, the medulla, or maybe both. And then also ask yourself, where are your arteries and veins? After you do that, start to tackle what are the different capillary beds. Where would you find them, with what nephrons are they associated with, and then ask yourself kind of what feeds those capillary beds and what's draining those capillary beds. So this is all of the information that we've covered in the past few discussions over the nephron. It looks really daunting when you look at it in one figure, but kind of look at this as you're going to break it down and look at the different areas, and then it's not going to seem as overwhelming. You should try to tackle the anatomy of your nephron and kind of have an idea of bare basics of their functions. Are they going to be involved in reabsorption? Are they involved in secretion? Are they involved in both? And if so, which one are they more involved with? Are they involved in filtration? Then kind of tackle worrying about, if you have those bare basics, moving into the videos going over the nitty-gritty details of how those components work. All right, so that kind of completes the macroanatomy and microanatomy of the kidneys. And in some future lectures, you're going to see in nitty-gritty detail how those nephrons work to produce urine. What we're going to move on to next are going to be the other organs that are going to be found in your urinary system. So the next one that we have to worry about would be the ureters. And the ureters are going to be these thin, long tubes that are going to carry urine away from the kidneys to be stored temporarily in the bladder. So these are going to start at about the level of L2, and it is going to be this thin, narrow tube that is going to be a continuation of the renal pelvis. So remember, the renal pelvis is going to be kind of the funnel that is wider, and it will carry urine into the thin ureter itself. The ureters are going to also be retroperitoneal. So they are going to be found behind the peritoneum, just like the kidneys. But then they actually have to enter into the bladder. How they enter into the bladder is they actually pass through the posterior wall. So they go to the back of the bladder and then enter into the bladder on the posterior wall of your bladder itself. You have a left and a right ureter, so you would have then the ureters entering into the posterior wall of the bladder on both the left and the right side. And when you have the bladder pressure increasing, you're filling up your bladder with more and more urine, the distal end of the ureters will actually close off so that you don't have any backflow of urine from the bladder into the ureters and back up towards the kidneys. So the ureters are going to carry urine down to be temporarily stored in your urinary bladder. And so the urinary bladder will also be retroperitoneal. The kidneys, the ureters, and then the bladder are all going to be retroperitoneal organs. The bladder is going to be positioned on the pelvic floor posterior to the pubic symphysis. So if you remember back to A&P-1, you had your left and your right os coxae that would come together to form a joint, the pubic symphysis, and so the bladder would be located posterior to that joint. It is going to be a muscular sac, and this sac can actually expand as the bladder is filled. There is a difference between the anatomy of your urinary bladder if you're looking at males versus females. So this figure right here on the left is showing you a male bladder. And you can tell that this is a male bladder because you have a prostate gland right here that is going to be just inferior to your bladder. And so when urine is draining out of the male bladder, it has to pass through the prostate gland before it continues through the urethra outside of the body in the penis. So in males, you have a kind of walnut-sized prostate gland that is going to be inferior to the bladder, and a portion of the urethra has to run through the prostate gland. In females, that prostate gland is absent, and you would find your female bladder located anterior to the vagina and the uterus. So the urine is going to be carried down to the bladder via your left and right ureters. Once the ureters reach the bladder, they are going to have urine enter into the bladder through these small openings called the ureteric orifices. So you have one on the left side and one on the right side. These openings are going to be found kind of at the posterior floor of your bladder. And you have one other opening. This is going to be the opening to the urethra, so it would be draining the bladder, and that is going to be the urethral orifice. And if you then connect the dots between the opening that drains the bladder, the urethral orifice, and the left and the right ureteric orifices, you are going to see that you kind of have this triangle. This triangle on the floor of the bladder is called the trigone, and it is going to be a smooth area. And since you have all of these openings kind of going to this area, this can be an area where if you have a bladder infection, the infections can kind of persist in this region. The urinary bladder is made out of smooth muscle. It's going to be a sac of smooth muscle. And the bladder is able to expand as it fills and then kind of collapse down to a smaller size if it's empty. When the bladder is empty and kind of at a smaller size, you are going to see rugae, or these kind of ridges in the walls of your bladder. The rugae, it actually looks almost like webbing, so it is pretty intricate, and this rugae will disappear as the bladder is filling and expands. The rugae will disappear and it looks smoother. When you have the bladder filling and expanding, it can actually rise superiorly in a pretty significant fashion. And this is going to allow for the bladder to expand, but then not have a significant rise in the internal pressure. That if you have the container kind of expanding and becoming larger, you're not going to have a buildup of pressure within your bladder. The bladder can expand quite a bit and can hold a pretty large amount of fluid. So a full bladder can be expanded to about 12 centimeters in length, and it can hold about 500 milliliters of fluid. If you are really filling the bladder, you can have the bladder hold twice that amount if necessary, but you do have the risk of, if the bladder cannot drain, that the bladder could burst if it's overdistended. So for whatever reason, if the bladder is unable to drain, the bladder would be able to expand kind of twice its normal expansion, but then beyond that, it could burst. Draining the bladder is going to be the urethra. So the urethra is going to carry urine from the bladder to the exterior. And there is a difference between a female urethra and a male urethra. In females, the urethra is very short. It is only about 3 or 4 centimeters. It's tightly bound to the anterior vaginal wall, and you would have the urethra opening to the outside of the body at an opening called the external urethral orifice. The external urethral orifice will be the opening that is going to be anterior to the opening of the vagina. In comparison, the male urethra is going to be much longer than a female's urethra. The male urethra is going to have 3 distinct regions. There's going to be one region that is draining the bladder, that is your prostatic urethra. So the portion of the urethra draining the bladder and then running through the prostate gland is your prostatic urethra. There's going to be a second portion that is pretty small. It's going to be just this region right here that is going to be passing through a layer of muscle. This is the membranous urethra, or the intermediate part of the urethra, that passes from the prostate to the beginning of the penis, and it's going through a very kind of thin layer of muscle. The third part of the urethra in males is going to be the spongy urethra. And this is going to be the portion of the urethra that passes through the penis itself. The spongy urethra is going to be what actually opens to the external environment through the external urethral orifice, which would be the end of your urethra right here. The male urethra also differs from the female urethra in that it is going to have dual function. It is going to be part of the urinary system because it's going to carry urine from the bladder and out, but it also is going to be part of the reproductive system because it will carry semen. So the male urethra is technically best thought of being a urogenital organ because it's involved in both carrying urine and semen.